Okay, very good evening to all the aspirants, very competitive aspirants of botany and plant sciences. Today's our eminent resource person, Dr. M. Sujatha Madam Garu, working in working in Government Degree College for Women, Autonomous, Karimnagar, as an assistant professor. Madam has huge experience, more than 30 years, like my teacher. Dr. Sujatha Madam Garu, his PhD completed in 1988. So that's the madam's experience. మన అందరికీ కూడా టీచర్తో సమానం. Dr. Sujatha Madam Garu, we are very lucky in this evening. Madam is going to explain about the hepatic opsida. That is also we can call as the liverworts. The liverworts, most important type study, that is the Marchantia. Madam is going to explain about the Marchantia, morphology, anatomy, reproduction, life cycle. Madam, please welcome and start your session. Madam, please welcome.
Okay, thank you, sir. Thank you very much. Good evening, sir, and dear students. Today, yesterday we have studied about that is Bryophyte's general characters and economic importance. In continuation with that, today we are going to study about the first type, that is bryophytes are divided into, as you know, bryophytes are divided into three groups. That is Marchantia, that is Hepaticopsida. Second one is Anthoceratopsida. Third one is Bryopsida. So we'll start with the first type, that is Hepaticopsida. Hepaticopsida, under that, the type what you're going to study is Marchantia. So first of all, what is Hepaticopsida? Hepaticopsida yesterday we discussed that hepatic means liver.
Liverworts.
Hepatic means liver lobe-like structures are present in the thallus of bryophytes. Such bryophytes are included under Hepaticopsida. Under Hepaticopsida, Rixia and Marchantia, these are the examples. But today we are going to study about Marchantia. Marchantia, its external structure, internal structure, that is anatomy, as well as reproduction, that is life cycle, is also included in this. So here you on this slide you are seeing the structure of Marchantia thallus. It is liver lobe like you're seeing. So about that you're going to study, and at the center you're seeing some cup-like structures that we'll study in detail afterwards. So coming to the classification, whenever you're studying any type, we should know about the classification of that. Based upon its characters, classifications are given. So classification of Marchantia, that is also known as systemic position. Classification is also known as systemic position. So it comes under the kingdom Plantae, and the division is Bryophyta. Under Bryophyta, it comes under the class Hepaticopsida, and under the class, it comes under the order Marchantiales. Order always ends with the suffix ales. So Marchantia name plus ales, if you add, that will become the order. So Marchantiales. And family, it is named after Marchantia only. Family always ends with the AC, that is a suffix. So here the family name is Marchantiaceae. The genus is Marchantia. I'll repeat it. Kingdom Plantae, division Bryophytes, class Hepaticopsida, order Marchantiales, family is Marchantiaceae, genus is Marchantia. Next is habitat, and that is where they occur. Habitat means the place where they are found. Here habitat as well as distribution you are seeing. Distribution means on the earth in which places these are available. Habitat, the place where it occurs. As you know, bryophytes, they are amphibians of kingdom, they can grow both in water as well as on land. So habitat, that is near the water bodies, it will grow. In moist places, they'll grow. So coming to the genus, under Marchantia, there are 65. There are so many genera. That is, Marchantia is the genera. Under that genera, 65 species have been identified. Where they'll grow means, as you know, bryophytes grow in cool places. So the best place they grow is cool, moist, and shady places. One of the species, Marchantia polymorpha. Polymorpha, it grows as a poiner in burnt forest soils. Poiner means wherever forest burning, if it takes place, then completely all the plants are burnt, but the starting that is poiner, the starting plant which can grow there when moisture is available, that is Marchantia polymorpha. So this is about the habitat. Then its distribution in India. We are studying about the, that is, its distribution means the places where it is occur in India. 11 species of this have been identified in the western Himalayan regions. Marchantia polymorpha, it occurs at high altitudes in Himalayas, on moist river banks and rocks. And another species, Marchantia palmate, occurs in Kashmir, Kumun, South India, Bengal, and Assam. All these are the cool places. Then another species, that is Marchantia similane, it occurs in Himachal Pradesh. So this is about the distribution. Next is about the plant body, that is the habit or the structure. Plant body, as you know, bryophyte's plant body is a gametophyte. Gametophyte will produce the gametes in the gametangia. So the plant body is known as gametophyte. It is always haploid. So here you're seeing, right hand side, you're seeing the plant body of Marchantia. It is thallus, and this thallus is bilobed, lever-like structure, and each is divided. The main thallus is divided into two branches, so dichotomously branched. And how it grows means it grows straight on the substratum, so it is called as prostrate. It is not erect; it grows flat on the thallus, so it is called as prostrate, and it is dichotomously branched. Here, dorsal side and ventral side of the thallus are different, so it is dorsi-ventrally differentiated. That in anatomy you'll understand. Dorsal side, in the diagram you're seeing the thallus, one of the lobe of the thallus which is divided into two branches. At the middle you're seeing a midrib-like region, and at the center of the midrib you're seeing the cup-like structures. So coming to the thallus, this is the dorsal surface of the thallus. Dorsal means upper surface of the thallus, a shallow groove marked by the presence of distinct midrib in each branch. Dichotomously branched, this is. Two branches are there. At the center, groove. It is not flat completely. Somewhat the thallus, it forms a groove-like structure. So at the center, you will see the midrib-like region. The blackish line, what you're seeing is the midrib. Many polygonal areas which demarcates the outline of underlying air chambers. Here, air chambers surrounding the air pores on the dorsal surface, just like stomata. Here, these plants are not well developed. So, instead of stomata for gaseous exchange and respiration, small chambers are present, small pores are present. That pores opens into the chamber. So, that is, there many cells are present in internal structure that you'll see in detail. Each polygonal area has a pore in its center called air pores. And along with air pores on the dorsal side, you'll see the gamma cups. the whitish cup-like structure in which green color structures you're seeing gemma. So that complete white
మమ్మీ
color structure is called as
కార్ కీస్ ఎక్కడ పెట్టినావు?
gemma cups. Gamma cups are present along the midrib. So that you're seeing in the diagram clearly. Each branch has a growing point. Here at the tip of the branch, dichotomously branched thallus is present. At the tip of the branch, in this diagram, you can see notch-like structure you're seeing at the tip. So that same thing is enlarged in this diagram. So each branch has a growing point situated at the apex in a groove called as apical notch. It is a notch like that is somewhat inner part is present, that is called as notch, and that is the growing part. That is cell division will takes place there. Whereas the ventral surface, lower surface of the thallus, it is here also dichotomously branch only you'll find, but on the ventral surface you're seeing brownish color patches you're seeing in the midrib like region. Midrib is like region is present upper side. On the exactly on the lower side you're seeing brownish color scales. Scales are present as well as rhizoids are present here. Rhizoids, they help in absorption of water, and different types of rhizoids will be there that you'll study in detail. So on the lower surface, you can see the rhizoids as well as the scales in the midrib region. About the rhizoids. In Marchantia, rhizoids are present, but there are two types. Rhizoids are usually unicellular and colorless. That's why in this diagram you are unable to identify the rhizoids. They are colorless. So scales only you are able to see. But the rhizoids, they are unicellular and they are colorless. They are made up of single cell and they are colorless. Two types of rhizoids are present. The upper diagram you are seeing smooth walled rhizoids. That is, the inner wall is smooth there, so they are called as smooth walled rhizoids. Whereas the lower diagram, tuberculate rhizoids. These rhizoids are called as tuberculate rhizoids because from the walls some papillae-like outgrowths will grow. That's why they are called as tuberculate rhizoids. In the diagram, it is clearly visible. Inner wall show papillae-like outgrowths. The function of these rhizoids is anchorage, means for fixation, as well as absorption of water and minerals. Along with rhizoids, in Marchantia, on the ventral surface, you will see that is the scales, the brown colored structures, what you have seen earlier diagram. So they are the scales. They are in the diagram you are seeing the scales, appendiculate scales. Here you are seeing many cells. They are made up of many cells. So they are multicellular. Scales are multicellular, but they are one cell in thickness. Single layer only they are present. So that is a one celled thickness of cells are present, and they are violet colored due to the presence of anthocyanin pigment. So the anthocyanin pigment in plants, it gives violet color. So also in Marchantia, scales, they are violet in color. How these scales are arranged means arranged in two rows on either side of the midrib. In this diagram you are seeing brownish color you are seeing, that is actually somewhat violet colored. Afterwards they'll turn into brown, but initially they are, when they are live, that is that time violet color. So two rows they are present. Then they are of two types. Scales are of two types. One is appendiculate scales, second one is ligulate scale. Appendiculate scale means the upper diagram you're seeing These have an apical subrotund appendage that is somewhat round and form inner row of scales. They are present toward the inner side of the that is thallus, toward on the exactly on both the sides of midrib. Appendiculate scales. Second one, ligulate. Ligule means tongue-like. The lower diagram you're seeing, that is a ligulate. Tongue-like scales, these are. The upper one is appendiculate. Ligulate, this is a small without appendage and form outer row of scales. Means at the center on the ventral side, in the midrib region, appendiculate scale. Beside that, on outer side, ligulate scales are present. What is the function of scales? Scales is it gives protection. So protection of the growing points and retention of water by capillary action. So these are the plants, they'll grow in moist places to retain the water. Scales, they help in, that is, they help in retaining the water by capillary action. Next, on the upper surface, there is the anatomy we are studying now, internal structure of the thallus. Earlier we have studied the external structure, morphology. That is. Now we are studying the anatomy of the thallus. On the upper side, thallus is differentiated. It is dorsi-ventrally differentiated. So dorsal side and ventral side of the thallus is different. It is not same. So also in the lower diagram, you're seeing that is upper side and the lower side are different. On the upper side, as you know, thallus is covered by epidermis. Lower side also epidermis will be there. Upper side also epidermis will be there. But on the upper side, epidermis will have certain pores. A diagram, you're seeing the pores. So these pores, that is, they're called as air pores. They enter into an air chamber. So these pores, what is the shape of this is barrel shaped. They are barrel shaped. Each pore is surrounded by 4 to 8 superimposed tier of cells. Means they cover one another, sometimes 4 or more, up to 8 number. That many this tier of cells are present to protect the pore from desiccation. Each tier consists of a ring of 4 to 5 cells. You are seeing here in this upper diagram 4 cells. The cells of the lowermost tier protect inwards, giving the pore a star-shaped appearance. So in the lower diagram, you're seeing the TS of the thallus. Upper diagram, just the pore. In the thallus, upper side and lower side are differentiated. The upper half of the tires project outwards and half project inwards. That is about the pores. But upper side, you are seeing photosynthetic region. In the next diagram, that description will be there. Next slide, description will be there. So photosynthetic region, lower one will be the storage region. So about the pores, pores have comparatively wide pore passage in the middle than the margins. Toward the margin side on the thallus, upper side, less number of pores are present, whereas at the middle, somewhat comparatively more number of pores are present. Now you are seeing the exact anatomy of that is the tears of the thallus. Upper side, that is the photosynthetic region. Lower side is the storage region. So about the photosynthetic region, the parts which are present in the photosynthetic region. First one is where it lies. It lies below the epidermis. As I told, thallus on both the side it is surrounded by epidermis. Upper epidermis and lower epidermis will be there. Just below the upper epidermis, this photosynthetic region you are seeing with the chambers. It consists of large air chambers. In the epidermis, you are seeing the pores. These pores are opening, they'll enter into a chamber-like structure that is called as air chambers. So many air chambers will be there on the upper side, and these in each air chamber, photosynthetic filamentous structures are present. So it consists of large air chambers separated by single layered partition walls. The partition you're seeing just beside the air chamber is written, no, that line you're seeing a wall like structure, that is a partition. Just like that other side also you're seeing the partition. So partition, that is air chambers, are surrounded by this partition. Inside this air chamber, you'll see the photosynthetic filaments. And these photosynthetic filaments, they'll have chlorophyll and they help in photosynthesis. That's why this region, upper region, is called as photosynthetic region. Lower side, you're seeing the storage region. After photosynthesis, the food material which is prepared is stored on the lower side for future use. So that's why it is called as storage region. Just below the photosynthetic region, you're seeing the storage region, and just below the storage region, a single layered outermost layer, that is lower epidermis, you're seeing. On the lower epidermis, usually in the middle region, you're seeing the rhizoids and scales. So coming to the storage region, it lies below the photosynthetic region, compact zone of several layers of polygonal parenchyma cells. All this lower side storage region, you're seeing so many polygonal cells. Polygonal means many angles are present, they are not round, there is no intercellular space. These are all parenchyma cells. They'll store the food material. Cells devoid of chloroplast, as as it is storage region, there is no chloroplast and no intercellular spaces. Cells contain starch and protein granules that is stored there. Starch and proteins. Mucilage and oil containing cells are also present inside this. Storage region, thick at the center and tapers toward the margins, because thallus is thicker, multi-layered at the middle, and toward the margin it is thin. That's why at the center it is thick. Cells of the midrib region are elongated with reticulate thickenings will be there. And on the lower side, from the lower epidermis in the midrib region, rhizoids and scales are seen. So this is the anatomy. Dorsiventrally